My name is Serghei Mangul. I'm from University of California, Los Angeles. It was a long flight, but I'm really excited to present our new method to profile immunoglobulins from the regular RNA-Seq data. So to get started, I want to just give a naive view of the adaptive immune repertoires as a bioinformatician. So if you think about the adaptive immune system, so that is encoded by immunoglobulin and T-cell receptor leukocytes present across several chromosomes. So if we zoom in into one leukocyte, so we will have a collection of the V genes, which is variable genes, D genes, diversity genes, and J genes, joining genes, which are actually recombined in the random fashion. So for example, in this cartoon example, you see gene V1, D3, J2 being recombined, and at the junction point between V1 and D3, for example, we will insert the non-template insertions. So the machinery, the sophisticated machinery of the immune system allows you to achieve the astonishing diversity with the estimated size of the repertoire, in other words, the number of sequences, to be 10 into power 16. So a couple of definitions we will need in this talk. So we'll be talking about the complementary determining regions, and there is 3 of those. The CDR3-1, which is within V gene, as well as the CDR3-2. But the most interesting one is CDR3, which is spanning the junction and includes part of the V gene, the entire D gene, and part of the J gene, as well as the non-template insertion. And then, since it's the most diverse one, so we will use that to define our clonotype as the set of distinct CDR3 sequence. And the collection of all the clonotype individual we will refer as the repertoire. Okay, so what, why, how, and then technology is currently used to profile the repertoire is the targeted technologies where you use the BCR-Seq or the TCR-Seq. So we want to use the regular sequencing, which we generate on the daily basis, and which is far more sequence data being generated comparable to BCR-Seq. So the challenge is that actually those immune reads, they will be among the unmapped reads, which is currently thrown away into trash. So we want to change that model from the picture like that into something like that, which is more suitable, I guess, better for the environment. Okay, so let's have another look, keeping the mapped and unmapped reads in mind, let's have another look at the immune repertoire. So in black, you see the mapped reads. So since the V, D, and J are part of your genome, so there will be no problem to map. If the read is entirely within those genes, there will be no problem to map those by any high-throughput mappers. So those are mapped reads. So what about the unmapped one? So those are in gray, and then it will be quite challenging to capture those. So there is 2 reasons for that. So first reason is if the read is kind of those guys, they will have extensive somatic hypermutations above the threshold of the high-throughput mapping tool, so they will be not captured. As well as this sophisticated case where the read is spanning the junctions, and then as you can see, the part of the read belongs to V, the part of it belongs to the insertion D, and so on. So that will be not captured among the mapped reads, and this is what exactly we will be targeting by our method. So the data sets, so we combined a couple of data sets. So we took in-house data, which was lung, nasal, and blood, as well as we took the whole GTEx data, which is up to 9,000 samples coming from up to 600 individuals spanning 53 different body sites. And then just generalized whatever we observed. We also took randomly 2,000 SRA RNA-Seq samples. So in total, we got 1 trillion reads, more than 10,000 samples, and at least 54 tissues. So let me talk a little bit about the method. And then the method is quite simple, and then there is a trick, which I will explain, which makes things quite trivial. So let's look at the read, which we need to convert into the protein sequence. And then we just need to know the definition of CDR3 sequence, which is the sequence which starts with the cysteine and ends with the phenylalanine. So keep this in mind. We look for such reads which will contain that sequence entirely within the read. So this is our read. Let's scan the read from the left until we find the first cysteine. That will be it. We scan the read from the right, and then we find the first phenylalanine. So as we do so, that's our candidate CDR3 sequence. However, that could be false positive just because we know nothing about the flanking regions. So what we do, we take the database of the V genes, which is quite small. There is around, in total, across the all chains of the immunoglobulin, there is around 500 genes, V genes. And then we just take one by one, and then we don't need alignment just because we match C by C and C here, because all the V genes, they will, the very last or couple of before last amino acids, they will be always cysteine. Okay, and then we just compare one by one the bases. And then we do so for every V gene until we find the match. So V2 didn't match, and then actually V3, that's your match. Okay, so we found the corresponding V3 gene, so we repeat exactly the same procedure for the J genes, and then let's say GA4 was your match. So that will be the CDR3 sequence the method will assemble or reconstruct. Okay, and then that's, yeah, so that's not anymore candidate CDR3, that's a CDR3 assembled by the method. Okay, so what about the reads are short? And then they will not span the entire combination, meaning that will not map both V and J. Instead, it will only map V or only map J. So let's try to rescue those reads as well. So that's your read, quite short. You find the cysteine by exactly the same procedure of scanning the read, and, but you were not able to find F. So let's don't give up, and then let's do the same procedure of looking for the V gene, and let's say we were able to find the corresponding V gene. That's great. That's our partial CDR3 sequence. Let's keep it. Let's keep all of partial CDR3 sequences where the read overlaps only V, and then the same, let's keep only those which overlap J, and then all we need to do, we just need to find the ones which come from the same CDR3 sequence based on the overlap. And then we use the suffix G for that. So that's your read overlapping on the V3 gene, and that's your read overlapping on the J4 read, gene. And then in case they do overlap and agree on the overlap, we require overlap of 5 to say that those two reads come from the same CDR3 sequence, and then we will be able to reconstruct the full CDR3 sequence. Okay, so we also think about the sequencing errors, which might be within the CDR3 region, and for that we use the clustering, where here every dot, that's a full-length CDR3 reported by the method, and then we do the clustering based on the pairwise eddy distance between the CDR3 sequences. And we report the consensus sequence for every cluster as a true CDR3 sequence, and we also ask for the support of at least 2 reads to CDR3 sequence to be reported. So we did some simulations where we see ImRep, that's the name of our tool, outperforms the existing methods, specifically in the recall rate, and also highlighting that our method is able to handle the very short reads, like 50 base pairs. If you want to kind of sum it up into the one measure, which will be the harmonic mean on the recall and precision, which is called F-score. So the existing, the best method existing so far, called MixCR, is able to achieve around 20% accuracy measured by F-score, and our method is able to provide 80% F-score on average. So we were happy with that. As well as we did validate the ImRep based on the real data, where we took 18 tumor biopsies sequenced by both BCR-RNA-Seq and RNA-Seq, asking the question how well RNA-Seq does compare to the BCR sequencing, which is kind of the proper technologies coming at the additional cost, as RNA-Seq, as doing what we do with RNA-Seq is kind of free of charge. And then what we were super happy to see is that the immune diversity, which was measured basically taking the number of your clonotypes, looking at the relative abundances of those and doing the Shannon entropy. So with the immune diversity reported by BCR-Seq and the immune diversity reported by the RNA-Seq based on the ImRep were very well correlated. So one caveat I need to disclose is that the BCR-Seq was done on DNA level, and then the RNA-Seq was obviously done on RNA-Seq level. So we didn't control the experiment, we took the publicly available data. So so the caveat of that is that sometimes in a couple of samples, we observe the main clonotype from the BCR-Seq not being expressed in RNA-Seq. So we did exclude those samples. As well as we asked the question of how much of the entire repertoire which we think the BCR-Seq captures, ImRep is able to get. And then we see that almost 70% of the entire repertoire ImRep is able to capture. And then also, since the reads are short, it's kind of in line what you saw on the previous simulation, we were able to reconstruct a magnitude more clonotypes compared to the existing method. So I think it's like one thing I want to mention, the key to success here was that that was the right tissue, and then what I say by right tissue, that was for us. That was a tissue very rich in B cells, so that was bone marrow, as well as the sequencing was quite deep, so it was 100 million reads per samples. They were short, they were 50 base pairs, but we were fine with that. Okay, so let me jump into the results. So running that across the entire GTEx data, we got 3.6 million different CDR3 sequences. And then the distribution across the you will see all the tissues, 53 different tissues across GTEx being on the x-axis, and then the clonality, which we estimate as the total number of clonotypes normalized by per 1 million original RNA-Seq being on the y-axis. So what we see, I guess, as expected, we see quite high number. And then one more thing I need to mention, that we split the clonotypes into 3 classes, each corresponding to each chain. So we have a heavy chain, IgH, and then we have 2 light chain, kappa and lambda. So we present those in the different colors. And then what we see as expected, I guess, that the secondary lymphoid organ like spleen and terminal ileum from the small intestine, they show a quite high clonality. We're surprised to see that the blood-associated sites didn't show much of the clonality. But I guess the most surprising thing was right here, where you can see those kind of stack of tissues. Those are the tissues which are rich in mucosa. And then, so minor salivary gland and colon, they show quite high immune clonality, even higher than the terminal ileum, which is the proper secondary lymphoid organ, which is in the GTEx was designed, which was designed as the proxy for the lymphoid organ, just due to the close proximity. So that can be indication of saliva, can be great tissue to study the immunity. So we're checking that right now. Okay, so we'll also ask the question, are those 3.6 million CDR3 sequences we did assemble, are they more like private specific, meaning present in one individual, or they're more public, and then also are they tissue specific? Okay, so, and then here you can see number of individuals sharing the particular clonotype sequence, and then number of clonotypes. So I guess as expected, the vast majority of the sequence we assemble, they are private. With the certain exceptions, so there is a certain number which are public, and those are the most extreme public sequence we did find in our data. So for example, for IgH, which is heavy chain, we found that particular CDR3 sequence, which was present 58 individuals. For the light chains, which are less diverse, they expected to be more public, and then we see a couple of sequences, CDR3 sequences, which is present almost the entire GTEx cohort. We also asked the questions, are there public clonotypes are more tissue specific? And then the answer is no. So we see the majority of the assembled CDR3 clonotypes being both tissue specific and individual specific. Okay, so we did create the atlas of the immune repertoire, which we call the AIR. It's available on the GitHub, so where so far we share the public clonotypes, and then we're planning to extend that and provide all the clonotypes. So if you're interested, check it out. So actually, that's the largest resource so far in terms both on the number of CDR3 sequences assembled as well as the number of tissue types being involved. So we also have the pathology nodes as the part of the GTEx consortium, and then as well as the histological images. So we did use that to study the Hashimoto disease, the autoimmune disease present in the thyroid. So as you can see, as the severity of the disease increases, the size of the repertoire, in other words, the number of clonotypes increases. So you can view that as a part of the validation, as well as we look at the breast tissue. Before that, we look actually at the sex differences, and we see none, I guess as expected. But in the breast tissues, we see quite significant difference between male and females, where, interestingly, for the male, we found the outlier known from the pathology nodes to have the gynecomastia. Hope I did it right. So where the male breast becomes like female. Okay, so if you're curious to know what about other unmapped reads, we have the answer for that as well. So we did the read origin protocol to explain almost all 99.9% of all the unmapped reads, and then the data set was the same, 1 trillion reads across different data sets. So details are on the archive if you're interested to check. Okay, so I think I'm ready to wrap it up and say that we took the GTEx data, the unmapped reads from the GTEx data, we ran the ImRep, and then we created the large collection of the immunoglobulin sequences. And then they will be available soon as the part of the atlas. All the details I was talking about, they are on the bioarchive in those 2 papers. And actually, as the future work, we plan to scale, possibly to profile all existing sequencing data, including whole genome, RNA-Seq, and single-cell data, with the aim to create the global immune repertoire atlas, like immunome, of all the nations and people in the world. And then if you have a data you want to share, be part of it, so please talk to me. So this is the people who are involved, in particular my PI, Elazar Eskin, and then our immunological expert, Maura Rossetti and Mark, and as well as the Igor Mandrik from Georgia State, who actually developed the tool, and GTEx Consortium and other people. So ImRep is available on the GitHub as the one-line solution. You can just run one line and then it should work. We preferably would like to have mapped and unmapped reads together. If not, we got a new feature. If you forgot to save the unmapped reads as part of your BAM file, we got you covered. Okay, thank you.
Are there questions? It's okay. So have you done this on the TCGA RNA-Seq data?
Yeah, so actually this is current work.
Because I thought it would be interesting to see if you can predict from that data whether there is a huge lot of neoantigens present in some of these tissue samples, because that could be used for deciding whether a patient should be eligible for immunotherapy.
Yeah, yeah, that's exactly on the list. So I think the goal is like from TCGA, just because the immune repertoire is such a sensitive thing, even compared to the gene expressions. So we will aim to predict the survival rate, and then, as you said, like the result of the immunotherapy. Just like kind of the final goal will be like to inform the person if it makes sense to make that particular immunotherapy.
Yeah, I think comparing that to, because some of these data have exome data,
yeah,
the mutational load from exome data. Because I'm still curious whether we can even figure it out, whether the patient has a lot of neoantigen or you just have a cold.
Yeah, yeah, that's a great idea, Francisco.
Hi, thanks for a great talk, and I apologize if you talked about this and I missed it, but so for this kind of superfamily, you will expect a lot of multi-mapping reads. What do you do with them, or you don't see them?
Can you clarify what's the reason for the multi-map reads? Because the CDR3 sequences, they're quite unique, and that will be determining our reads. So I don't quite see the reason for multi-map reads.
It's been my experience that sometimes the CDRs are similar, but if you don't see it, that's
yeah, they will be similar, but they will be similar. That just mean that we have multiple reads supporting the same CDR3 sequence, so that will result in the frequency being higher of that particular clonotype.
Oh, I see. I see what you're saying. Thank you.
Thank you. Nice talk. Two questions. So the first one, what is the best sequencing depth for RNA sequencing to capture the diversity? The other question, how does your method deal with the novel alleles? Like, for example, in your case, I think you consider clonotypes that have, even if they might have different V genes or like few SNPs in the V genes, so this might produce completely different clone. But it seems to me that your method is still consider this as one clonotype. So can you please comment on that?
Yeah.
Thank you.
So yeah, it's a great question. So the answer to the first question is like, I think as much as, as more is better from the depth. I guess we currently exploring. So we got this 100 of million reads, so we want to subsample it and see how much you will lose in terms of diversity. So I don't have a formal answer now, but I guess you can think of that as the very rare component of the sequencing. So increasing the depth doesn't have a linear effect, because usually the number of reads coming from the immune repertoire is quite small. So we will have a formal answer, I hope, soon. And then coming to the second question, yeah, so I think it really depends what you define a clonotype and how you deal with the somatic hypermutations, I guess. And then so far, and then there is such a variety in the literature, people thinking about 80% identity to 100% identity, and then consider the alleles as a separate clonotype or like within the same. So our definition was just the same amino acid sequence. So if there is two CDR3 sequence with the distinct amino acid sequence, we'll call them different clonotypes.
All right, can I ask another question?
Sure, please.
So in terms of the tissue, so the tissue you used in your experiment was bone marrow, was it?
Yeah, so the bone marrow was used for the validation, and I guess that's the best tissue for RNA-Seq, just because the portion of the B cells among the reads coming from the B cells receptors among the total reads is the highest.
So do you think it would work if we sequence blood tissue or PBMCs? Or?
Yeah, blood will be not that great, just like you can just go to this plot, and then that's kind of will answer your question where the blood is quite low. But that's again number of distinct CDR3 sequencing, otherwise clonotypes per 1 million reads. So that's not great. Maybe saliva actually. This is what we're trying to investigate. Maybe saliva will be the best available or easy to get tissue to study the immunity.
Okay, so I'll chat to you after that. Thank you.
Thanks very much again. And we're going on to the next session led by